The conference is about to begin. Please take your seats and switch your mobile phone to silent mode. Please be reminded to keep your mask on throughout the conference. Thank you for your cooperation. Ladies and gentlemen, good morning. Welcome to the Future Science Prize and the University of Hong Kong Joint Academic Conference. I'm Michelle Young, a fourth-year medical student here at HKU. First of all, I would like to invite Professor Max Shen, Vice President and Pro-Vice Chancellor Research at the University of Hong Kong, to give his welcome remarks. Professor Shen, please.
Thank you. Ladies and gentlemen, good morning. What a happy day for Hong Kong U. I would like to welcome all of you to this joint academic conference on behalf of the University of Hong Kong. We're here today to honor two distinguished scientists from the Li Ka-Chin Faculty of Medicine, Professor Yuan Kuo-Yung and Professor Joseph Sriya Malik Paris. They have been awarded the 2021 Future Science Prize in Life Sciences for their contribution in pathological discoveries in Sars and its zoonotic origin. Their seminal discoveries have helped government and public health agencies to develop more effective response strategies against emerging infectious diseases such as COVID-19. Their dedication and scientific rigor represent the best of Hong Kong U and remind us that human ingenuity is the key to the world's most pressing challenges. The Future Science Prize was established in 2016. In just a few short years, this privately funded award has become one of the most prestigious science prizes, not only in Great China, but also in the world. And this prize is also a reflection of the tremendous advancement in research and development taking shape in this region. We are deeply honored that this year, two of our own faculty members have been recognized for their scientific leadership, which has made a positive impact worldwide at such a critical juncture. We hope that this award not only serves as recognition for many years of research excellence, but also inspires generations of young scientists to carry the torch and advance science for the betterment of humankind. So I just want to remind you that today we have a lot of exciting talks and roundtable. There will be a roundtable discussion, the present and future pandemics following the keynote speech. We hope you will enjoy the discussions with our panel of experts. Once again, congratulations to Professor Yung and Professor Paris. Thank you.
Thank you, Professor Shen. Now we'll play a video message from Professor Dong Xin Lian, Rotating Chair of 2021 Future Science Prize Life Science Subcommittee, for her welcome remarks.
Good morning, everyone at the University of Hong Kong. First, let me introduce myself. I'm Xin Lian Dong, the Rotating Chair of 2021 Future Science Prize Life Science Committee. I'm an investigator of Howard Hughes Medical Institute and an arts and science professor of biology at Duke University. Today, we're here to celebrate the work of Professor Kuo-Yung Yuan and Professor Malik Paris from the University of Hong Kong, who won the 2021 Future Science Prize in Life Science for their discovery of SARS-CoV-1 as the causative agent for the global SARS outbreak in 2003 and its zoonotic origin. Professors Yuan and Paris' work had major impact in our ongoing struggle in combating COVID-19 and emerging infectious diseases. During the outbreak of severe acute respiratory syndrome, also known as SARS, in 2003, Professors Yuan and Paris's team treated the first SARS patients in Hong Kong and isolated SARS-CoV-1 from their clinical specimens, which was critical for the design of diagnostic tests and disease characterization. In addition, Professor Yuan's continued studies on SARS-like viruses in wild bats greatly increase our knowledge of zoonotic reservoirs, barrier to cross-species transmission, pathogenesis, and clinical diagnosis of these viruses. Because of the high prevalence of SARS-like coronavirus in bats, the discovery predicted the potential reemergence of a SARS-like epidemic and stressed the importance of public health preparedness. Before I hand the floor over to our two proud winners, I would also like to introduce to you the Future Science Prize, which is the first privately funded science prize established in mainland China in 2016, with 3 award categories, including life science, physical science, and mathematics and computer science, to recognize scientific breakthroughs in mainland China, Hong Kong, Macau, and Taiwan. There are no restrictions on citizenship. In fact, Professor Paris is the first non-ethnic Chinese winner for the prize this year. The selection process for the winners takes several months of very intense work involving nomination, which is by invitation only, so no self-nomination is allowed. The review and final decision-making is done by a scientific committee of distinguished scientists. For each candidate, the scientific committee will conduct the international review process for the originality and impact of the work. The winner is then determined by unanimous vote of the entire committee. So we try to keep the process as unbiased as possible. In fact, none of the Life Science Committee members work in the same field as our 2 awardees this year. But this did not stop us from recognizing Professor Yuan and Professor Paris's work. Through awarding those who have made great contributions to the society, like Professor Yuan and Professor Paris, we hope to make them the role model for young people, to spread the spirit of science, to encourage the pursuit of scientific careers. In my case, I would like to encourage women female students to pursue scientific careers and to transform the future with science. Congratulations, Professor Yuan and Professor Paris, for your seminal contribution to our understanding of emerging infectious diseases from SARS in 2003 to current COVID-19, which led to more effective responses and strategies in controlling these diseases. Lastly, I wish today's Future Science Prize Symposium a big success. Thank you.
Thank you, Professor Dong. Now may I invite the moderator, Professor Avian Flu H5n1, Ru Shen and Helen Lei Professor of Health Sciences Pedagogy, Chief of the Division of Infectious Disease in the Department of Medicine of HKU. Professor Hong, please.
Thank you, Michelle. So good morning to you all. It's my real privilege to be here. And I would like to express my heartfelt congratulations to Professor K.Y. Yuen and to Professor Monique Pieris. And I have the privilege and honor to be working under the 2 professors from 2003 SARS all the way to the latest COVID-19. So may I now invite Professor K.Y. Yuen to give the first keynote speech from SARS 2003 to COVID-19. Professor Yuen, please.
Well, thanks to Professor Ivan Hong's kind introduction and also Professor Dong's introduction too. And thanks to the organizing committee for organizing this both on-site and also the online conference. Thanks for coming to support me. Well, my lecture is from SARS 2003 to COVID 2019. So if you use the word coronavirus to do a PubMed search, between 1951 to 2002, there are only 1,800 publications on 10 coronavirus only. Now, from 2003 to 2019, there is another 11,000 publications, already more than 60 novel coronavirus. But now, within just 2 years, we have more than 0.1 million publications, much more on COVID-19 and SARS-CoV-2, of course. And 300 of them is from the University of Hong Kong Department of Microbiology, and more than 30 novel coronaviruses are named after Hong Kong U. We love Hong Kong U so much that we try always to name them after HKU. Now, so let's have some introduction on coronaviruses. So by 2003, we have alpha, beta, and gamma coronavirus, and with a similar genome arrangement of ORF1ab, spike, envelope, membrane, and nuclear protein. Now, if you look at phylogenetically, you can see that the IBV, the infectious bronchitis virus, is discovered in 1937 as what we call a gamma coronavirus genus. And then later we have the mouse hepatitis virus, which is a beta coronavirus. Now, by 2003, we have 10 coronaviruses, including 2 human coronavirus, which cause common cold. And one of them is a 229E belonging to the alpha coronavirus. Now, of course, things change a lot during the 2003. We have the SARS-CoV-1. Now, I deliberately use SARS-CoV-1 to differentiate from SARS-CoV-2, which is causing the COVID-19. And that has markedly disrupted the socio-economic activity in Hong Kong. And of course, if you look at the airport now, it's just as empty as in 2003, showing that emerging infectious disease do have a lot of force, a lot of entrance. So what happened in 2003 is that we saw and worked up the first cases that is coming into Hong Kong SAR. And from one of them, actually, the lung tissue biopsy grows this SARS-CoV-1 virus in pure culture. Together with Malik Pires and also our team members, we not just isolate the virus, because isolation of virus was also successfully done by 2 other groups in US and German. What we also do is to prove the disease association with SARS-CoV-1. That is, we use the patient's profiles of viral load and the specific antibody response to show that that is indeed the case, but not in the control patients. Now, with the availability of RT-PCR test and also the specific antibody test, our team members, Professor Yi Guan and also Professor Bao Jinjian, they are able to hunt down that the civets in the Shenzhen wildlife market indeed contain a very similar virus with 99.8% nucleotide homology to the human's SARS-CoV-1. And that is very important because control of infectious disease is control at the source. No, but, well, unfortunately, mainland scientists subsequently show that it's only the market caged civet having the SARS-CoV-1 infection. We can't find any virus in the wild civet or those in the civet farms. And that means that the market civets are only intermediate or amplification host. So there must be another identified animal, which should be the natural reservoir of SARS-CoV-1. Now, so the key question is, where is it? Where is that animal? Now, so literature review first always, and you know very well that many of those emerging viruses, like the Lissa virus, which is related to rabies, the Hendra virus, the Nipah virus, the Ebola and the Marburg virus are found in bats. And that is a very important source of information. And therefore, we focus on hunting for bats, sampling them throughout the rural areas in Hong Kong, We sampled 11 locations during summer to winter. And then that is important because at 2 locations, we did found a SARS-related coronavirus in the bats. And what we use is very simple. It's just taking the specimens and then do consensus RT-PCR followed by whole genome sequencing of any virus that we found. So this is one of the positive locations, which is a water tunnel, and the bats are all hanging on the roof of the tunnel of caves in the rural area of Hong Kong. Now, in winter, it's very simple because we just use a net and a stick to dislodge these bats into the nets for sampling. Now, remember, during winter time, they do not fly, they're hibernating. That makes life very easy. And of course, it's a collaboration with Mr. Sheck in Agriculture, Fishery, and Conservation Department. We collect them, we measure them, weigh them, and then sample the oral cavity and anal canal. Usually the palate of feces that come out when the bat is very frightened. That is an excellent sample. And of course, in summertime, it's much more difficult because the bats are flying. So we have to set up all this mist net along their pathway of flight. So they fly onto the net, being caught below the net, and then start collecting, sampling, and measure them. So this is the natural reservoir that we believe to be carrying the ancestral virus. We call it bat SARS-related coronavirus HKU3. And very interesting small animals with a thickened piece of skin, look like horseshoe around the nose and the mouth. Now, the most important issue is up to 39% of them, the specimen are RT-PCR positive for this bat SARS-related coronavirus. And they're roosting on the cave and tunnel top in hundreds, allowing the rapid dissemination and amplification of the virus among the bats. And of course, there are other bat species which would allow basically the transmission across species. And our team members, very important, have been hunting for these novel viruses for the last 18 years. Patrick, Susanna, Kelvin, Herman, and Sid. So without them, all these could not have been done. Now, after we found the HKU3, the bat SARS-related coronavirus, another very the energetic group, which is Professor Xijing Li at the Wuhan Institute of Virology, they also discovered even closer bat SARS-related coronavirus in Yunnan. And that is very important. It adds a lot of information because some of it is very close to the SARS-CoV-1 and also the SARS-related coronavirus in bats. And if you look at this phylogenetic tree, it's obvious that the civet, the human coronaviruses, actually with the Xijing Li's virus, they actually come very close together with one immediate common ancestor, suggesting that only one interspecies jumping event, maybe from the bat to the civet and then to human, or from bats directly to civet and human. And that is very interesting. Unlike the bat SARS-related coronaviruses that we found in Hong Kong and others in Yunnan, they are of polyphyletic origin, not from one immediate common ancestor. And these are from different species of horseshoe bats, not just Rhinolopus sinicus, the Chinese horseshoe bat, but other species. Now, with all this important finding, actually, in the year 2007, which is 4 years after SARS, we write a review article in the Clinical Microbiology Review. Now, in the last paragraph on should we be ready for the reemergence of SARS, we actually write this. The presence of a large reservoir of SARS-CoV-like viruses in horseshoe bats, together with the culture of eating exotic mammals in southern China, is a time bomb. The possibility of the reemergence of SARS and other novel viruses from animals or laboratories, and therefore the need for preparedness, should not be ignored. Now, so we predicted it in 2007. We've been waiting for it, asking for government funding to continue to hunt it, and we believe that further surveillance virus hunting program would be very strategic in preparing us for the next epidemic. Now, so we have setting up many different sampling sites from the rural area of Hong Kong together with the AFCD, in Guangdong, in Guangxi, in Yunnan, where there is the highest animal biodiversity, and to Hainan. Actually, Hong Kong is not tropical. Hong Kong is only subtropical. The Hainan is the place where the only truly tropical area in mainland China, and also to Bangladesh, and also in Dubai, and up to Ethiopia. So that is the sampling sites where we collaborate with them and get these bed specimens and other specimens for our virus hunting program. So with these hunting program in the last 15 years, we have discovered more than 80 novel viruses. 30 of them are novel coronaviruses in our department. And of course, some of them have clear public health significance. At least the Chinese saucer bat as a natural reservoir of SARS-CoV-1 is highly likely. And then by 2017, which is 10 years after we discovered the bats, coronavirus HKU2, Xi Jinping's group report mainland outbreaks of fatal swine diarrhea in piglets due to HKU-like coronavirus. So we did have found something which has some public health significance. Now, but then some of the government funding agency actually commented in our reports to them that, well, KY, you have found many animal coronaviruses, but very few of any public health significance. And I'm very worried because when they say this, it means that the funding is going to stop. Now, I'm a very lucky person because one morning I read the New England Journal of Medicine in the year 2012, and that is what Professor Ron Faucher writes in a brief report for New England Journal of Medicine about the MERS coronavirus, which is the Middle East Respiratory Syndrome coronavirus. And they found this MERS-CoV, and in a summary, he said, the closest known relatives are the bat coronavirus HKU4 and HKU5. Now, I immediately sent this paper to then the Secretary of Food and Health, Dr. York Chow, and our funding was continued. I'm very lucky. Now, so this is a lesser bamboo bat, which is very small, talking about 4 cm in length, living inside the hollow of bamboo. And it is carrying the bat coronavirus HKU4. And this is the Japanese Respiratory bats, which is carrying the bat coronavirus HKU5. And of course, after so many novel viruses that we found, we never really appreciate the significance of what we found in the year 2005, the SARS-related coronavirus in Chinese saucer bats. Not until 2019. So we have to wait for a long time before something happened. Doing research is like this. You never know how important and impactful of that finding may be many years later. So we have to be really patient and have some luck when your funding can be continued. Now, so what happens is that my colleagues and myself are actually working in the Hong Kong U Shenzhen Hospital. Now, in the year 2019, by December 30th, which is actually my birthday, I heard from my contacts in mainland China, academic context, they say that something is happening in Wuhan. And that it's like SARS. Now, and then very soon, the mainland scientist, Professor George Gao, actually, and his team and other members and the collaborators, they announced that they have found a new coronavirus, which is similar to the SARS-CoV-1 and also bat-related coronaviruses. And they then published the first genome, allowing the public to know about it. And at the same time, we found a patient coming into the Hong Kong U Shenzhen Hospital on the 10th of January. It is part of a family cluster of pneumonia at the Hong Kong U Shenzhen Hospital. You can see the CT scan of lungs showing multifocal peripheral ground glass changes. And what is more important is that we published at the same time, they published in New England Journal of Medicine, January 24th, and we published in Lancet. Showing the same thing, that the SARS-CoV-2 coronavirus is clustering with other bad SARS-related coronaviruses. But they are on a different branch with the human SARS-CoV-1 in 2003 and the Paguma civet of 2003. So both find the same pattern. It's a different virus on a different branch of phylogenetic tree, but they are all related to bat SARS-related coronavirus. Now, so this family cluster is interesting. This is a family cluster of 7 people, a 7-member family. They have traveled from Shenzhen to Wuhan two of the family members has visited a hospital in Wuhan, and then they fly back to Shenzhen, and then presented to us with persistent respiratory symptoms, and then we found the other family members are also infected. Now, one of the family members stayed at Shenzhen, never left Shenzhen, did not go to Wuhan. She was also infected and developed the symptoms later, well, which likely is because of the other family members transmitting the SARS-CoV-2 to her. Now, more importantly, 3 of the family members have very mild symptoms, or 1 have no symptoms at all. So this is the first documentation of person-to-person transmission of SARS-CoV-2 leading to COVID-19. Half of them have mild or no symptoms, and that have a lot of pandemic significance. Now, so you can see that COVID-19 is quite different from SARS in 2003, because the 2003 SARS patients, they have abrupt onset of fever, respiratory symptoms quite severe usually, and also radiological changes of pneumonia. And they are rarely asymptomatic. Basically, everybody is symptomatic. We only found 1 case of asymptomatic shedding of SARS-CoV-1. And the viral load of SARS-CoV-1 actually peaked at around day 10 after symptom onset and then start to go down. When the viral load goes down, it coincides with the rising antibody in the patient's serum. Now, so that is quite different from COVID-19 because Dr. To, our head of department, he did a very important piece of research published in Lancet Infectious Disease. He showed the daily self-collection of posterior oropharyngeal secretions, now called early morning deep throat saliva, 深喉唾液. By clearing the throat 5 times before breakfast and mouth rinsing, he showed very clearly that the actual viral load of COVID-19 patients peak at the time of presentation or at symptom onset. And that is very important because it shows that it's not easy to control it because the viral load already peaked. The patient may have transmitted infection before you even know about it. And of course, the next question that we are asked in the laboratory is, why is that so? Now, we know that once a virus, an RNA virus, enters the cell, the RNA genome will be sensed by the toll receptor 7 and also the RIG-I. And that leads to a vigorous interferon response, usually. But the SARS-CoV-2 viruses produce a lot of proteins, structural and non-structural and accessory proteins, and quite a number of them significantly impair the interferon response, as demonstrated in our transfected cell line luciferase reporter assay. And that may explain why the patient's viral load can go up very rapidly and only becomes symptomatic when the viral load peaks. Now, with this information in the test tubes, we have to prove it at least ex vivo. So in this case, we're using what we call ex vivo lung tissue explant culture. And we ask patients who have lung cancer after the operation to donate some of the normal lung tissue to us. And then we do virus challenge. And they show here very clearly that the interferon response and also the interleukin-6 response, which are antiviral and inflammatory response, are markedly impaired in SARS-CoV-2 when compared with SARS-CoV-1. So SARS-CoV-1 is more inflammatory, and that may explain why COVID-19 patients, the initial symptoms are very mild, and there's early peaking of viral load. Well, with this important laboratory information and ex vivo culture information, Professor Ivan Hong did a randomized control trial using a triple combination of 3 modestly active drugs with the interferon beta 1b as the backbone drug. And it shows clearly that with this triple combination, if given early, it reduces the viral by 2 to 3 logs between day 6 and day 11. Now, subsequently, another paper published in Lancet Respiratory Medicine also showed that lentivialized interferon beta 1a in another randomized control trial show quite a bit of clinical improvement if it is given early. And that is actually very similar to influenza because, say, for oseltamivir, an antiviral for influenza, is only really effective if it is given within 40 hours after symptom onset. Now, the importance of interferon response in the control of SARS-CoV-2 is also illustrated by another 2 science papers. They show very clearly that those with severe COVID-19, they have autoantibody blocking the interferon alpha and omega. and some others also show that the deleterious genetic variants or loss of the toilet receptor 7 would lead to severe COVID-19. But there is one very important point. The most important underlying diseases, hypertension, cardiovascular disease, commonly found in elderly and obese patients. What is the role in severe COVID-19? That is still unexplained. So another very important report, also around the time of COVID-19, published in Lancet, it showed that the plasma ACE2 level correlate with deaths due to heart failure, stroke. And that is very important because ACE2, is a receptor for SARS-CoV-2. And then very soon there are papers coming out showing that plasma's ACE2 level predicts the severity and outcome of COVID-19 patients. Now then, we do another important experiment and actually show that, confirm that there is an enzyme called the ADAM17, which is a protease, can split the cell membrane ACE2, release as soluble or secreted ACE2. And this soluble ACE2 can bound, of course, onto the SARS-CoV-2 spike, and then this can be uptaken by the angiotensin II receptor type 1. Or the patient's own serum vasopressin can bind to this ACE2 spike virus complex and then taken up by the arginine vasopressin receptor 1b and then go in by receptor-mediated endocytosis, leading to the infection of cell. Now, this finding is very important by Philip. Why is it so important? Because it shows that even the lung tissue, the airway tissue, actually with quite a low amount of ACE2 when compared with the kidney cells and also the intestinal cells, they can still be infected very well. Because not just the cell membrane ACE2 is important, the soluble ACE2 is also important. So in a way, the SARS-CoV-2 exploits the vasopressin renin-angiotensin system for cell entry. And that also explains why higher plasma soluble ACE2 in patients with high blood pressure and cardiovascular disease develop more severe COVID-19. Now, this is what we are still doing. And you can see that once the virus enters the cell would mount antiviral response by two mechanisms. And one of the mechanisms, of course, is the interferon response, going through the interferon regulated factor 3. The other response is to activate the caspase so that the cell undergo apoptosis and thus stop the virus from replicating. Now, what we found is that actually, when the caspase 6 is being activated, it can cleave the viral nucleocapsid protein into two, and it bound onto the interferon regulatory 3. And that lead to stop the interferon induction and then allow better virus replication. And we show that inhibitor of caspase 6 can actually reduce the virus replication. In a way, this finding is important because we show that the SARS-CoV-2 exploits two host antiviral mechanisms, the apoptosis pathway through the caspase 6 against the interferon pathway through interferon regulatory 3. So you're exploiting two host antiviral mechanisms against each other so that the virus can grow better. So after so many years, 18 years, we have found many coronaviruses. Now, in the gene bank, you have more than 100 coronavirus species already, of which 34 coronavirus are from the University of Hong Kong. We actually have defined within the beta coronavirus another lineage called the lineage D, the HKU9. And we also defined a new genus of coronavirus, which is the Delta coronavirus. In the past, nobody knows that there are Delta coronavirus genus existing. So we found another 13. Most of them are from birds. And now we have sufficient genome information to tell us about the evolutionary model of coronaviruses. And that, we believe, is very important for years to come for coronavirologists. Now, so this is our proposed evolutionary model. We believe the bats and birds are the coronavirus ancestor natural reservoir. So you can see here that for the alpha-beta coronaviruses, a lot of them are from bats, and that such bat coronavirus cannot be found in the case of the Gamma and Delta coronavirus. It shows that the bat coronaviruses are the gene pool or the gene source for the generation of alpha and beta coronaviruses. Now, similarly, these are the Gamma and Delta coronavirus. They are all from mostly are from birds. And that doesn't happen in the case of alpha and beta. There is no bird coronavirus here, suggesting that the bird coronaviruses are the gene source for the generation of Gamma and Delta coronaviruses. And because of these differences, it is understandable that for alpha and beta coronavirus, the interspecies jumping event leading to important public health events, such as the rhinolophus bat HKU2 virus jumping into swine, causing farm outbreaks of swine diarrhea, leading to a lot of piglets dying, is one of the events inside the alpha coronavirus coming from bats to pigs. And of course, we know that now it is very likely the bat coronaviruses, which are SARS-related, may have jumped from the bat to an intermediate host and then to human, causing the SARS in 2003 and also the COVID-19. And there is probably an unidentified lineage C bat coronavirus, which is jumping into the camels and human, which is still causing the MERS outbreak continuously in the Middle East. And one very recent report, which is a preprint online by a group by Linda Safe in US, showing that the porcine Delta coronavirus HKU15 has jumped into human in Haiti. And that actually is very closely related to the sparrow HKU17. Now, so I just want to impress upon you that we are humbled by these viruses. We have learned hard lessons from SARS, MERS, COVID-19, and influenza. And it seems that many of them, the ancestral origin is from the viruses in the bats and birds. And I must say again that bats and birds are natural reservoirs for many viruses, including coronaviruses. They contain the resident viral gene pool for the evolution of novel virus species by mutation and recombination before they jump into other animals. They are warm-blooded vertebrates with unique genomes and high metabolic rate. They have unique immune system tolerating the virus infection. The bats are all asymptomatic, for example, although they have a lower body weight compared with the non-infected bats for the bat SARS-CoV-related coronaviruses. Their high species diversity, they roost together, they fly, they migrate, which facilitate virus amplification, transmission, and genetic changes and evolution. And that's why surveillance of wildlife, patients, animal and human interfaces are important for the early control of emerging infectious diseases and pandemic preparedness. So give us funding for further surveillance. Thanks to all my past and present principal co-investigators, especially molecularists and others, our scientific officers and graduate students, our clinicians in the hospitals and microbiologists, Agriculture Fishery Conservation Department, FEHD, Ocean Park, our mainland and international collaborators. So thank you very much for coming to support us. Thank you.
Thank you, Professor Yun, for the very lovely talk, unfolding the story of SARS 2003 to MERS and lately to COVID-19. May I now invite our second laureate, Professor Molly Perez, to give the second keynote speech, Emerging Infection from Bird Flu to COVID-19. I understand that Professor Perez is currently under quarantine after returning from US due to the very tight infection control measures imposed on our return travelers in Hong Kong. So I understand that Molly will be joining us online. So please welcome Professor Molly Perez.
Thank you, Ivan, for that kind introduction. And I would like to start by thanking the panel of the Future Science Prize for recognizing both KYUN and myself. But I think essentially they're recognizing our whole group who have been working for over 25 years. So if I can share my screen first. Yes. So I would like to go back a bit before SARS and start with avian flu, because the lessons that we are trying to learn are about these emerging virus infections that keep on crossing species from animals to humans. So I arrived in Hong Kong in 1995, but by 1997, we were confronted with the avian flu H5n1. If you remember, there were 18 human cases, 6 deaths, and we were very busy diagnosing and characterizing the clinical features of that disease. But the source of human infection was identified to be the live poultry markets, and with the slaughter of all poultry in Hong Kong, that particular outbreak was brought under control. However, after that, we realized that this virus is probably, although under control in Hong Kong, has not disappeared. So I joined my colleague Iguan and Ken Shortridge to carry out surveillance in the live poultry markets, wild birds, and also pigs in Hong Kong. And sure enough, we could see evidence of this H5n1 virus. And we could also see that it was initially back in geese. Then we can see it reassorting. Influenza viruses have 8 gene segments and changing species from geese to ducks and then back to chicken. So clearly we could see that this was coming back and going to pose a major threat again, zoonotically, to humans. So as part of these studies, we also were looking in detail as to how this virus is getting about, how it is being maintained and being transmitted. We were able to show that what really happens is that very occasionally the virus enters from the poultry farms very infrequently, but because many poultry from many sources are mixed in these wholesale poultry markets and come into these retail poultry markets, once a virus enters the retail poultry markets, it never leaves. Because if you imagine the situation where every day you have new poultry coming in, which are non-immune, and poultry being removed, this is an ideal situation for the virus. And we were able to prove that when the only way that you could break this cycle of maintenance was by closing the market completely for a day, the so-called rest days that were introduced in 2001, 2002. And here you can see a low pathogenic influenza virus as an indicator. Before the rest day, you have maybe 4 or 5% of birds infected. After the rest day, it goes down to 0. But then the next month, it is up again, and then it comes down again. So clearly showing that the virus is being maintained and amplified in these live poultry markets. And then subsequently, when there were outbreaks in poultry farms, we could show, in Hong Kong, we could show that the source of infection of these poultry farms was cages and people moving the virus from these retail poultry markets back to the poultry farms. So with this understanding, evidence-based interventions were progressively introduced in Hong Kong. And most recently, after 2008, when every night the market was kept empty of poultry, you can see the very dramatic impact it has on reducing the persistence of influenza viruses in avian influenza viruses in these markets. So as a result of this progressive surveillance, proactive surveillance and interventions, no further cases of severe avian influenza have been reported or detected in Hong Kong since 1997. However, of course, in 2004, many countries, regions, reported the presence of H5n1 outbreaks. And because of our work, we had been designated a WHO reference laboratory for H5 viruses, and we were able to be involved very much in countries such as Vietnam, Indonesia, training people, and also, for example, the very first human case of H5n1 in Indonesia was confirmed in our laboratory. Indeed, the first case was detected when I was on a training course in Indonesia, and then the confirmation was carried out in our laboratory. Now, more recently, with H7N9, the studies from mainland China showed that, of course, the live poultry markets were a risk factor, but there were some people who developed disease with H7N9 who had been to these markets, but not actually to the live poultry market. They had not bought poultry, they had not touched poultry at all. So together with my colleague Hui-Ling Yen, we asked the question, is this virus present in the air? And indeed, she was able to show using these air samplers that infectious avian influenza viruses can be detected in the air. And in fact, in particular, the highest concentration of infectious airborne particles was found in the vicinity of this defeathering machine. If you are familiar with the operations of live poultry markets, after killing the bird, it is put into this apparatus which spins at fairly high speed and clearly generates aerosols. So in collaboration with colleagues at the Faculty of Engineering, Dr. Yen was able to make some minor modifications to this defeathering machine to reduce the spread of aerosols. This is the existing defeathering machine, and this is the one with minor modifications. So just illustrating that, understanding how these viruses transmit, we can intervene to reduce the risk of the jump occurring from birds or animals to humans. Now, going further, by understanding the evolution of these avian influenza viruses, H7N9, but also H5N1, H10N8, they all follow a common pathway. As I reminded you, influenza viruses have 8 gene segments. So in all of these zoonotic viruses, 6 of these gene segments come from a very common poultry chicken virus called H9N2. But the H and the N gene segment comes from wild bird viruses. And these viruses do not, of course, directly transmit viruses to chicken, but they transmit viruses to domestic ducks. And because domestic ducks and poultry are marketed in the same wholesale marketing system, this gives the opportunity for reassortments to take place so that you have continuous new zoonotic avian influenza viruses emerging. So if we can separate the marketing of terrestrial poultry, such as chicken, quail, pheasant, from aquatic poultry, that would be another way of reducing the chance of these emerging zoonotic viruses arising in the first place. Another aspect that I was quite interested in is why H5N1 causes such severe disease. Essentially, about 50% of patients who diagnosed with H5N1 globally die. So over the years, with a number of studies that we have reported, we showed that the gene expression profile, particularly the innate immune expression profile elicited by H5N1 in alveolar epithelial cells or in macrophages, is very different to that elicited by seasonal influenza, the common influenza that regularly infects us. And we identified the innate immune pathways that are dysregulated, including the TNF signaling pathway, the type 1 interferon pathway, JAK-STAT signaling, the apoptotic pathways, and COX-2 pathways. And also that these differences are quantitative rather than qualitative. So in order to try to understand the pathogenesis of this more clearly, my colleague Michael Chan and ourselves, we looked to develop a a model of the air sacs that you get in the lung, because this is where the pathology arises in severe viral infections. And essentially, what Michael was able to show is that when H5n1 viruses infect these alveolar epithelial cells, they downregulate sodium and chloride transporter channels that remove the fluid that continually leaks into the alveolar fluid space. So when these pumps, if you like, are blocked, you end up with accumulation of fluid in the alveolar spaces, and that is what leads to the acute lung injury. And Michael went on to show that using one type of intervention, which is mesenchymal stromal cell therapy, you can reverse the loss of these ion channels. And in fact, in animal models, you can improve survival of these animals. But mesenchymal stromal cell therapy, which, by the way, is currently being used in some centers for treating COVID-19, but it's quite a cumbersome approach. So we are now looking at other small molecule therapies to reverse this fluid accumulation and to reverse this the blockage of the ion channels, if you like. Then, of course, moving on to the 2009 pandemic, this emerged in Mexico, and very quickly, because of our studies which had been ongoing with swine influenza viruses, we were able to show how that pandemic virus emerged. And that was by the reassortment of two swine influenza viruses, one being what was called the North American triple reassortant virus, and the other one Eurasian avian-like swine viruses. But then the question is, until that time, people had assumed that swine influenza viruses are quite static geographically. So the Eurasian avian swine viruses essentially believed to be in Europe, the North American triple reassortant to be in North America, and the classical swine lineage to be in China and parts of Asia. But because of our work over many years, we were able to show that what was happening was not what people expected, that these swine influenza viruses actually do move across continents. So this is what the situation in China was. This is based on our surveillance in abattoir in Hong Kong, which gets pigs from about 10 provinces across China. So initially, there was this classical swine H5n1 virus in pigs. But by about 2001, you could see that this Eurasian avian virus had entered the Chinese pig population. And then 2002, the triple reassortant North American virus had entered the pig population. And clearly, what was happening was that because of importation of pigs for breeding, improving the breeding stock, some of these viruses were entering and moving across borders and being present in places that they were not expected to be. And of course, when you have different lineages of swine viruses, they would mix and match their genes. And indeed, that was happening as well. There was reassortments taking place. And one of these had 7 of the 8 gene segment combination that was found in the pandemic virus. Now, of course, phylogenetically, the pandemic did not arise from here. It arose in Mexico. But what we said is that these viruses are not separate by continents. What we were demonstrating in Hong Kong and China was probably happening elsewhere in the world, including in Mexico, and that is how the pandemic started there in Mexico. But it took 7 more years, in fact, for them to find the exact precursor of the 2009 pandemic virus. Now, as the pandemic spread worldwide, that virus, which came from swine, of course, went back into pigs across the world. And we were the first to report that after going back into pigs, the pandemic virus, the human pandemic virus, was now reassorting with the existing swine influenza viruses in pigs. So if you look at the landscape of swine influenza in Asia, the emergence of the pandemic has changed this landscape completely. If you remember, I told you there was the Eurasian lineage, the North American lineage, and now the pandemic getting introduced into pigs. And you can see that now, essentially in this region, not just in China, Hong Kong, but also in Vietnam, most of the swine influenza viruses have multiple human pandemic origin gene segments in them. In other words, they have very well adapted gene segments that are very well adapted to humans in these swine viruses. And indeed, at least one particular lineage, the human population immunity to that particular lineage is quite low, and therefore it does pose a pandemic threat. Now, I come back to this image and this particular virus, which was, as I told you, heading towards pandemicity, or being very similar to what happened in Mexico for the emergence of the 2009 pandemic. And as I told you, that the precursor was not found for many years later. Since we had these viruses, if you like, the building blocks of the potential pandemic, my colleague, Dr. Yen, used the ferret model, which is an experimental model to look at transmissibility, a model of transmissibility of influenza viruses in humans, to show that the pandemic virus, as expected, transmitted very efficiently by air in ferrets. The other swine viruses that I showed you before, they did not transmit by the airborne route. So it suggested that they didn't have the capacity to transmit efficiently from human to human. But this is that unique virus that I showed you that had got part of the way to pandemicity, that had modest transmission by the airborne route in ferrets. So then she went on to put in the missing gene segment, which was the neuraminidase from the pandemic, into this virus, and show that that addition of the neuraminidase gene segment indeed gave full transmissibility in the ferret model. And then with a whole series of other experiments, she was able to show that the receptor binding profile of this virus was similar to the pandemic, but it was the addition of the neuraminidase that was essential to release the virus from infected cells. So you can see that that original swine virus that I was showing you, the virus replicates, but it gets stuck on the cell surface because the balance between the hemagglutinin and the neuraminidase is not optimal for the virus to replicate and be released to spread to infect other cells. So it is the balance of the hemagglutinin and the neuraminidase that was essential for the virus to be released to be efficiently infectious and transmissible. Then with the pandemic, of course, we also collaborated with our epidemiology colleagues, looking at transmission within households, showing that the severity of the pandemic 2009 virus was very similar to seasonal flu viruses, and also that the viral load profile was very similar. But then we also carried out population-based seroepidemiology studies, which showed that within 5 months, you can see, this is the antibody prevalence, particularly in children. Within 5 months, 40% of children had been infected with this pandemic virus. We heard about it first in April 2009, and you can see by September, October, about 40–45% of children have been infected. So just showing how quickly these viruses spread. The other question, of course, is what was the severity profile of the 2009 pandemic? Now, if you just look at hospitalized cases, we are only looking at the tip of the iceberg, and a lot of the transmission that is taking place is under the water, the bottom of the iceberg. And the only way you can assess that is by doing seroepidemiology studies. So this we did, and together with Ben Cowling and Joe Wu and others. And what we can show here is this is the infection attack rate in the population. If you look at the blue dots here. So as I said, children were the most heavily infected and becoming less and less with age. And this is because very fortunately, because the older population had been exposed to a virus fairly similar to the 2009 virus. The 2009 virus, in fact, comes from the Spanish flu H5n1 virus. So the older population had been infected with viruses more closely related to the 2009 pandemic, whereas the children were not. So the infection was taking place primarily in the younger population. But if you look at severity, adjusted for the number of infections taking place, if you look at deaths or you look at ICU admissions, and this is a logarithmic scale, you can see that it progressively increases with age. So we were quite fortunate that the older population were partially protected because of this cross-immunity that was present. Otherwise, the 2009 pandemic also could have been quite significant in terms of severity, because you can see that if you get infected as an older person, your risk of dying or getting into ICU is exponentially higher. Now, we also were interested in trying to understand how to risk assess these animal viruses for pandemic threat, and receptor binding to the cell is crucially important. And John Nichols, Michael Chan, and others developed these ex vivo culture model where we can actually take human tissues and see how efficiently any animal virus can infect and replicate in these respiratory tissues. And using that, we can show that, for example, here, this is bronchus tissue that, of course, the seasonal influenza viruses replicate very efficiently. These are the brown dots that you see here, infected cells. H5n1 does not efficiently infect the conducting airways, and that is why its transmission from human to human is pretty poor. H7n9, in fact, does infect much better, and in fact, more people have been infected with H7n9. And H9N2, another virus, also can infect the conducting airways better. And now, of course, quite more recently, we are hearing about H5N6, and that's also a virus that can infect the bronchial epithelium. So we are concerned about that. Now, some of the other more basic studies that we were doing was looking at the mechanism of influenza virus genetic diversity, or what is called this quasi-species. Now, influenza viruses being RNA viruses, it is not one single sequence that you get. You have a swarm of mutant viruses in any patient or any individual. So a PhD student, Peter Cheung, was able to identify one particular amino acid mutation in PB1, which reduces the error rate in the polymerase of the influenza virus, polymerase, and the error rate in mutation rate. And interestingly, what he was also able to show is that when the error rate of the virus is reduced, meaning the diversity of this quasi-species is reduced, the virulence of the virus also is reduced. So these are reverse genetics engineered virus just with one amino acid difference. And what this amino acid is primarily doing is reducing the mutation rate in the influenza virus. You can see that these low error-prone viruses are much less virulent for mice than the wild-type virus. And of course, this also has applications in vaccine development, for example, particularly in live attenuated vaccines, to prevent the vaccine reverting back to virulence. Then, of course, we come to the story of SARS, and KY has summarized that to a large extent. As he mentioned, we were able to culture the virus, and then John Nichols was able to show this by electron microscopy to show that it was a coronavirus, and Leo Poon was able to obtain partial genetic sequence to show that it was a completely novel coronavirus. And with that information, we had the tools then to provide diagnostic tests, and within a week, we were providing diagnostic tests for Hong Kong. So essentially, providing the diagnostic test while continuing to refine and optimize and validate the diagnostic tests. And with this, we were able to show a number of characteristics of this disease. For example, the fact that it was present not only in the respiratory tract, but also in the stool, in the feces of these patients. And that became quite important when the Amoy Gardens outbreak took place, where that big outbreak was believed to be caused by aerosolization of infectious species. And the viral load profile KY did show, and this was quite important, as he mentioned, because it shows that the virus is less infectious in the first few days after onset of disease. And epidemiological data also suggested that transmission is quite uncommon in the first 4 or 5 days after onset of symptoms. In addition to that, together with Gabriel Long, in the Amoy Gardens outbreak, we were able to show that in the case of SARS, the vast majority of patients develop severe disease. Very few of them have mild or asymptomatic infection. Whereas with many other respiratory infections, such as influenza, you have the majority of them having mild or asymptomatic infection. And it is these two factors that led to the ability to contain and control SARS, so that by July, essentially, the WHO was able to say that all known chains of person-to-person transmission have been interrupted. But with influenza, there is no possibility of doing it because it is infectious from day one, or in fact, even before onset of symptoms. And indeed, with COVID-19, very quickly, we collaborated with colleagues in Guangdong CDC to show that the viral load profile in COVID-19 was similar to that of influenza. And indeed, we reported by early February that the transmission pattern was likely to be more similar to influenza than to SARS, so that the measures that were successfully used to control SARS were unlikely to work in COVID-19. And how SARS emerged, I think KY has mentioned, and Guani and Jembogen identified the virus in these civet cats, and almost certainly the closure of those wild game animal markets at that time prevented the reemergence of SARS from that reservoir, because even after the human transmission had been contained, it would have emerged from this reservoir if that had not been contained. And the bat origin KY has described. Now, coming to COVID-19, as soon as we heard about this outbreak in Wuhan, and the features that were resembling SARS, Leo Poon, in fact, designed primers that spanned the bat coronavirus group, and using SARS coronavirus as control, we essentially had a diagnostic test, which was rapidly published and was one of the earliest diagnostic tests available on the WHO website. And in fact, over 170 laboratories in 77 countries received reagents from us in the very early days to make their first diagnosis of COVID-19. Now, of course, we were quite interested in transmission of respiratory viruses because we knew that this was a big knowledge gap. We really knew very little about how respiratory viruses are transmitted. And that's why we set up a research program, a team-based research funded program to look at transmission of respiratory viruses. And one of the studies that was done, coordinated by Ben Cowling, was to look at the shedding of airborne particles from patients with respiratory infections. Influenza was our main target, but some of these patients did not have influenza. They had the seasonal coronaviruses. And we could show really that airborne droplets, both large respiratory droplets and very fine aerosol particles, were shed from the breath or from the coughing of these patients. But very importantly, also show that wearing of surgical masks essentially reduced and blocked the release of these airborne particles. So this information became extremely important in COVID because once it was known that there was transmission from pre-symptomatic and asymptomatic individuals, this provided some of the basis for the universal use of surgical masks to reduce transmission. And very finally, I want to come back to a different coronavirus, MERS coronavirus. KY mentioned this, initially discovered in 2012, and we were involved in also showing that the camels were the source of human infection. But more importantly, over the last number of years, we have been looking at camels found in a very big part of Africa, because over 75% of the world's dromedary camel population is not found in Saudi Arabia or the Arabian Peninsula. They are found in Africa. And wherever you find dromedary camels, you can find evidence of MERS. So the virus is present predominantly not in Saudi Arabia, but in Africa. So why has been no zoonotic MERS in humans not been reported in Africa? So I'm just summarizing a lot of studies that have been done over many years. Firstly, showing the prevalence of the virus in Africa, and showing by antibody tests, yes, you cannot find evidence of infection in even in camel-exposed people. But if you use specific T-cell responses, we can demonstrate that about 1/3 of camel-exposed individuals in Nigeria in this particular study were getting infected with MERS. And also, when you look at the virus sequence, you can see that the viruses in Africa are genetically distinct from the viruses in the Arabian Peninsula. And then we have gone on to show that these viruses in Africa have somewhat reduced replication competence in these ex vivo cultures of human lung, which I showed you before. But, and also that it is the spike protein that confers this difference in replication competence for MERS virus in Africa compared to the Arabian Peninsula. But it's only a very few amino acid differences that are defining this, this potentially reduced virulence. And we really need to draw attention for the need for enhanced surveillance of MERS coronavirus at the animal-human interface across the dromedary camel regions in Africa. At the moment, nobody is paying any attention to this. And I think it illustrates the importance of studies at the animal-human interface, as KY was saying, the importance of studies, not just in humans, but at that animal-human interface, so that we can be better prepared for future epidemics and pandemics. So what I've tried to show is from studies of influenza, from studies of SARS, and from studies of MERS, the importance of studying and surveillance at this animal-human interface so that we are better prepared for future pandemics. And of course, many people have contributed to this work. This photograph was taken soon after the 2003 SARS discovery. So you have some of the people involved, of course, KYUN, Iguan, Leo Poon, K.H. Chan, John Nichols, and then over the years, many others who were involved in the collaborative work that I have discussed, and a lot of work that I have not discussed. And of course, I want to pay tribute to Dr. Robert Webster, who was very important in supporting our studies at Hong Kong U. Thank you very much.
Thank you, Professor Malik Pierce, for giving another lovely presentation, highlighting the importance and the constant threat of both zoonotic and also human, not just coronavirus, but also influenza. And again, very, very important just to remind you to get your influenza vaccination as well as your COVID-19 vaccination. So we're now moving on to the roundtable discussion, present and future pandemics. And in addition to the two laureates, prize laureates, we have four very prominent scientists joining us today on the roundtable discussions. Joining us online, there will be Professor Shibo Jiang, who is Professor and Director, Institute of Medical Microbiology, College of Basic Medical Science, Fudan University. And also Professor Lin Fa Wang, who is Professor and Director, Emerging Infectious Diseases Program, Duke National University of Singapore Medical School. And here with us, of course, we have Professor Dong Yan Jin, who is Clara and Lawrence Falk Professor in Precision Medicine, School of Biomedical Science, HKU, and also Professor Liu Poon, who is the professor and head of Division of Public Health Laboratory Sciences, School of Public Health at the HKU. In the interest of time, what I will do is that I will be directing each of the questions to one of the panel members, and then if we have time, we will allow the other panel members to come in and join the discussion. And unfortunately, we will not be able to entertain any Q&A at this meeting. So may I now invite Professor K.Y. Yuen, Professor Ying, and also Professor Poon to join us on the panel discussion. Okay, so for the first question will be, how would COVID-19 pandemic turn out? And I think this is probably the most sought about questions. And I would like to direct this question to Professor Lin-Fao Wang online. Professor Wang, are you with us? Yes, very clearly.
Thank you, thank you. I mean, this is a billion-dollar question, so I'll do my best to try to paint through the crystal ball, I think, So I think it's clear that, even look at where Singapore is, the vaccine definitely play a very pivotal role to prevent, mass deaths and severe disease, but it's not good enough to prevent infection. So I think for us to exit the current COVID-19 pandemic, I think we needed to at least two things. One is the public health measure that the international community need to work together and get the vaccine distribution more evenly. I think that we need to reduce the burden of the transmission. Otherwise, I think just with the developed nation vaccinated, we're not going to be able to exit and prevent it. Secondly, is why we're doing this. I think scientifically, I think we still have to aim high to get at least a booster vaccine that is more efficacious. I think that we have three issues we're facing right now. The longevity is not as good as we want, and the spectrum, the breadth of the vaccine protection. For example, the current vaccine is not as well protected against the Beta and the Delta. And lastly is, I think more and more people realize that although the vaccine is very high antibody in the systemic, serum level, but the mucosal immunity may be the area that need to be improved. So if we, on one hand, with WHO's guidance and using public health and the international community work together, on the other hand, for the scientific team to come up with a better booster vaccine, I think we need both to get out the current pandemic. And in terms of time frame, I cannot predict. I think maybe another year or two. Thank you.
Thank you, Professor Wang. Professor Pieris, any comments on this?
I think I agree with Lim-Fa, and I think the crucial thing is the mucosal immunity. I mean, in fact, late last year, Professor Gabriel Young and I wrote a commentary to the Lancet exactly saying that although at that time there was all this optimism with the good results from the early trials of the vaccines that were coming out, that yes, it looks good for protecting against severe disease, but we cannot assume that it will protect against transmission. So I think we need a second generation of vaccines that will induce better mucosal immunity, and that is crucially important. But I think you also have to accept that this virus is not going to disappear. It's going to become an endemic virus that will be with us forever. But the aim would be to reduce the severity, the morbidity, and the mortality of this illness, so that it is no longer a fearsome disease.
Thank you, Professor Pieris. Professor Jiang, any comment on this?
Yes, about first of all, I will thank the committee invited me to give a chance to talk about my opinion. And first of all, I would like to congratulate Professor Yuan and Pieris for getting this as a Future Science Award. They really deserve it because their great contribution to the identification of SARS-CoV and other SARS-related coronavirus. I also learned a lot from this presentation today. But regarding the question about how COVID-19 would turn out, I agree with about professor's prediction, but I have a total different of concerns. Because many people believe that the virus will become weaker and weaker pathogenesis or transmissibility. So eventually will become flu-like coronavirus, like OC43 or 2009E. But I'm not so worried about the SARS-CoV-2. My father mutated into escape variant that's under the selection pressure of the antibody in the COVID-19 infected patient and also in the vaccine. Also, there's the drug will be coming to the market. And this drug may also cause the mutation of the virus, such as a new drug is called molnupiravir, just recently approved by the British Drug Administration Office. And they say this drug, it is clinically can reduce 50% of the mortality. And it is really great. But this drug will cause mutations in the virus, so to kill the virus because of mutation. But some virus might survive even with this mutation and become more pathogenic. So now the medical group now is investigating what's the future concentration about this mutant on the virus entry and virus replication. And so indeed, also we look back forward in order to look forward. It is not hard to invent a case where SARS-CoV-2 might further evolve into a nucleate like SARS-CoV-3 or MERS-CoV-2 by genetic recommendation with MERS-CoV. So we know that SARS-CoV-2 and MERS-CoV are conquered prevalent in the Middle East. So their genetic recommendation may happen upon the co-infection of both viruses, like an infected patient or the camel. Since both viruses can infect the same cells, like the type 2 elevated cells, and use identical transmission regulation sequence. So it also predicted that the receptor of SARS-CoV-2 possibly can bind into DPP4, the MERS receptor, based on a bioinformed and analyzed combining the human virus proteins interaction and the prediction of the protein domain. So if this happened, so this will be as a tragedy to the human peoples, because there's also is several cases of SARS-CoV-2 and MERS-CoV co-infection is already reported in the Saudi Arabians. So if this virus will become a new SARS-CoV-3 or MERS-CoV-2, they may maintain the high transmissibility of SARS-CoV-2 and the high mortality of MERS-CoV. So this will become very dangerous for the future. So that's one of the I'm very worried about.
Thank you, Professor Zhang. So maybe we move on to the next questions. The second question is, what should be done now to shorten the pandemic and reduce the damage, given the frequent emergence of variants, the lack of vaccine in developing places, and the vaccine hesitancy in developed regions? Can I direct this question to Professor Liu Poon, please?
Right. So I think we have to be practical and realistic. So history tells us that basically there's only one example that vaccine can able to eradicate the disease, smallpox. And then, but we do also know that there are a lot of vaccines which can effectively manage the disease a lot, and which can help to improve the public health in a greater extent. So we do have this example, and then COVID-19 vaccines now, yes, it's not perfect. Like we do have escaped breakthrough infections, but the disease, the severity and mortality rate is still a lot because of the vaccine. So I think we need to just try to disseminate evidence for local, like the vaccine hesitancy. So we need to disseminate the fact that, look, what we found from this vaccine is. So, and we have to use more innovative method to disseminate this knowledge and to multiple groups, stakeholders, so that they can understand the risk and benefit behind it. And then the other problem, which is I think is greater than vaccine hesitancy, is about the vaccine inequality. Right now, there's basically, I mean, just Professor Pierce and Professor K.Y. Yun said, they're like animals, they're reservoir. They can always spill over to human and cause disease. Now, if we don't manage to vaccinate people at a global level in a more equal manner, then we basically just allow the virus to circulate in a particular regions in a great extent without a controllable manner. So that is a problem, because that means they can always introduce the disease to other countries. I mean, Hong Kong, nowadays we don't have not many local cases, but then we have a lot of import cases. So we cannot keep things like this. So I think we need to try to increase the vaccine productions and also allocate these vaccines to other developing countries, and also the cold chain and other technical support, which is the developing countries to do at the moment.
Professor Jean?
So I agree with Professor Liu Poon, and I do feel that the work leadership and also the collaboration and cooperation between the different countries are really important in making more better vaccines, and also in educating the general public to receive the vaccines. And many of these are not only the task for the scientific community, but also for the work leaders. So it's actually crucially important that we can actually come together to come up with a roadmap to achieve some of these goals.
Thank you, Professor. Professor Yun?
Fully agree with what has been said, and I think the priority is now that everybody get vaccinated 2 doses, and then next generation mucosal vaccine. I declare my conflict of interest because I have dual influenza vector, the vaccine carrying the RVD. Now, and the third, I do believe is what is being designed by the group led by Professor Shibao Jiang, because they have a pan-Sabeco vaccine. I think that's maybe important in the long run, because if you look at the amount of SARS-CoV-related bad coronaviruses, the burden is high. And therefore, there is importance. If you can vaccinate the population, not just protecting against SARS-CoV-2, but other Sabeco virus, that might be very important in the long run. But I'm talking about in order you receive your 3 doses of vaccine first for the SARS-CoV-2, and then mucosal reinforcement, and then finally a pan-Sabeco vaccine. I think that's a strategy that I would see as important in the long run.
Thank you, Professor. So we move on to the next questions. The question is, why international governments fail to take lessons from bird flu and SARS? And I would like to direct this question to Professor Molly Pieris, please.
Yes, so I mean, I think this has been a source of a major frustration, because as Professor Yun also said, the warnings have been given over and over again about a number of these emerging virus threats. Now the problem is, of course, if you leave it to the current model is that we rely on the pharmaceutical industry to provide countermeasures, whether it be vaccines or antivirals. Now, of course, it's quite unlikely that a pharmaceutical company is going to invest huge amounts of money, because it takes huge amounts of money to take any of these therapeutic or vaccine interventions, from the drawing board all the way to market. They're not going to invest that amount of money without knowing that this is going to be a threat, that there's going to be a market. So this type of development really has to be taken on funded by other means. So I think it is very important that we take on board, at least at this late stage, the fact that emerging infectious diseases, particularly emerging viral infections, are going to be a continuing threat. And in fact, because of things that we humans are doing, the changes that we ourselves are making to our environment, they are going to be a continuing threat. And we need to come up with a different model to fund the development of these countermeasures, vaccines and antivirals to these emerging virus threats. Over.
Thank you, Professor Pierce. Professor Wang?
Yeah, I think that question is kind of a number 3, number 4. I think it's like the two angles of the same question. So maybe I can try to say that maybe we're good to answer in both ways. I think currently COVID-19 taught us 2 lessons. One is that the international collaboration, right? In Wuhan, general things, and 3 days later, the city was shut down on the 23rd. And then I think, there was a lot of discussion, and even to the level, I think there's a violation of human rights, a lockdown a city. And then if you look now 18 months later, some cities has been locked down 10 times. So there was obviously the international community was not ready to work together and to agree on a consensus. I think that need to be changed. Secondly is I think, as scientists, we're always thinking that science can do everything and anything. I think it's not. I think the political will, the diplomatic relation, and the legal framework, I always thinking that, the pandemic preparedness, science may play 50% of the role. The other 50% is the non-scientific. And that's the part I think is really lagging. And I don't have a solution, but I think that I can point out to say that's the area really we need to do much better. And third point I try to make is for any infectious disease, Merrick basically mentioned that the countermeasures and the private, industry cannot take the full burden. But I think same with the early warning and the prevention, So I still think that proactive surveillance, in the ICU units of different nations and make the data transparent and to do early warning, early prevention, plus the countermeasures, then I think it will be in a much better place. So it's a very complicated scenario, but I think we need to start from now, really do things what I call during peacetime, right? Before we hit another pandemic. Thank you.
Thank you, Professor. I think culture also very, very important with each different society. So let's move on to the next question. What should we do to prepare for further pandemics? And I would like to direct this question to Professor Jiang.
Thanks. Yeah, just like Dr. Wang said, I would combine the number 3, number 4 together. Actually, start from 2015, after the MERS COVID get into the South Korea. So through that time, I always published papers and to call for the development of the pan-cervical virus or vaccines or therapeutics and or pan-beta coronavirus vaccine and therapeutics. But unfortunately, I could not get any grant and I could not get any financial support from the pharmaceutical companies. They all said that you're simply too complicated. What we worry about? So this is SARS is gone and the MERS is already controlled. So that time I said maybe the future maybe have a more pathogenic virus will come out in the futures. But unfortunately, even government and in the pharmaceutical company and nobody is really pay attention to that. So just like in the 2003, after SARS came out, so many companies jumped in and to develop the vaccine and the therapeutics. But however, after just 5 months later, and all the companies withdrew decided this SARS might never come back. So I'm still in the beginning of last year. I also submitted several grants and talked to many companies because we already have a pan-coronavirus therapeutics candidate. Like we have the peptide that is against all the human coronavirus. And I asked some people to support our development for the clinicals. And also we already have the pan-cervical virus, which are against all the SARS-CoV-2 and its variant, and also against the SARS and the SARS-related coronavirus from bat. And it's very good. It's very potent. The neutralizing antibody is more than 10,000 against this virus. But unfortunately, so nobody's interested. They said this virus might not mutate and this virus might be gone and in a few months, just like SARS. So, and I told them that this is lucky we can get a vaccine, the first generation vaccine in less than 1 year, but it's less than 2 years. But in general, the vaccine development is more than take about 30 years. So like a SARS vaccine, AIDS vaccine is already 40 years, but we still don't see any hope. However, why the SARS-CoV-2 vaccine can be developed in such a short time? Because this coronavirus contain the fatal toxicity cell receptor binding domain, the RBD. The RBD, any vaccines contain the RBD, like they can induce antibody. But unfortunately, the first generation of the vaccines all contain the mostly weak the immunogenicity of the vaccine, either inactive virus particles or the spike proteins. But the best, based on our experience, the best vaccine should be RBD dimer, RBD trimer, and which can induce antibodies more than 100-fold higher than the spike protein, than the inactive particles. So they can induce antibody can have the provide 50% protections easily. But most of this vaccine have a common weakness just because their use induce antibodies very low. So like in the human, where the first generation can only induce antibody titer is less than 300. So after 6 months, they will reduce also less than like 50s because the half-life of this antibody is very low. It's very short. It's only about 3 months. So every half year, they reduce about 4 times. So that's why the first generation antibodies can induce the 50% of the protection in the peak time. But after 50 years, they will almost have no effect against particles, the virus variant. So that's why we also hope we should develop a pan-cervical virus or even pan-beta coronavirus vaccine against the currently and the future emerging and re-emerging coronavirus infections. Thanks.
Thank you, Professor Jiang. Professor Poon?
Right, I think now when we talk about future pandemics, when we look back, the 2009 pandemic is very mild. SARS, we can control it. So, I think now there was, before COVID, there was a perception that maybe we can control outbreak disease better than before. But in fact, now COVID-19 just turned out to be another case. And to be honest, the mortality rate right now is only about 2%. So think about MERS, H5n1, H7n9. The disease mortality is actually very high. So if you have a pandemic like this, that would be disaster. So I think now the governmental leaderships to have to appreciate that. One, the money that they spend on R&D is tiny in compared with what they have spent in controlling the COVID-19 right now. So I think we have to build up the further better infrastructure to do preventive measures, have to build up pandemic preparedness plan. Not many countries have that, right? And then how different government can work together to exchange information. I think that would be very important for facing the future challenge of the next pandemic.
Thank you. Professor Ching?
So actually, each pandemic comes with a slightly different way. So we cannot use the same method to tackle everything. For example, SARS-CoV-2 is very different from avian flu. So some of the errors that the authorities make at the start of the pandemic is to deal with SARS-CoV-2 using ideas that are derived from combating the avian flu. And it is also slightly different from SARS-CoV-1. So actually it is important that we actually make some long-term investment to support fundamental research, basic research in the broad field of virology and infectious disease. Over.
Yeah, so very, very important in terms of the translational from bench to clinical to bedside. So coming to the last question of the roundtable, what would be the most likely microbial agent for the next pandemic? And I'd like to direct this question to Professor Yuen.
That seems to be too difficult. And therefore, I just want to go back to the other two questions. That would be easier for me. I think the problem is that, especially the Western world, the government changes every 4 to 5 years. And the leaders will say, hey, pandemic, it only happens every 10 to 40 years. I would take my bets and not to deal with it. And then after a pandemic, well, I would spend some money, but then very soon I would shift all the money to other priorities, economic recovery, poverty, etc. So every time, even for Hong Kong, all the opinions that we give after 2003 SARS, many are not being carried out. And that is not complete and reasonable because there are always competing priorities. Now, so one of the ways to tackle this issue is that any candidates running for political leaders must have the recipe, curriculum vitae of having COVID-19 hospitalized, going in the ICU, the more severe, then you should be elected. Then they won't forget it like Mr. Boris Johnson. Well, joking aside, let's go back to question 5. So next microbial agents or next pandemic. Very difficult to answer, but you look at scientific evidence, what pandemic has happened before. If you count all those viruses that we have, remember, many of them may have jumped from animal to human, like measles. Even maybe chickenpox is jumping from animal to human. So any viruses that is currently circulating, any genus may have a possibility of jumping into human and then causing a pandemic. Now, but if you look at what has happened, we have very effective insecticide. Now, so arthropod-borne viruses, I think, is relatively less likely to cause a pandemic, despite global warming, because you generally have quite effective insecticide. Now, the other, of course, is you have very effective sewage system and clean water supply in many parts of the world. So the chance of diarrheal diseases due to emerging viruses are less likely. And then you think about it, the most important is actually respiratory viruses. Very difficult. Unless there is other dimension emerging, the chance is that the next pandemic would still be a respiratory virus. And to me, it seems that the literature shows that influenza, the most important cause of pandemic, recorded in human history in 1918, 1957, 1968, and then 2009. So the chance that that is important. We have to think about it. Again, the work by Professor Marek Peres in Hong Kong University have been focusing on this, and I believe that would continue to be important. But I would tend to feel that Peter Palazzi is also correct in saying that if it is a pure avian virus, the chance of it adapting to human is perhaps questionable. Now, but look at what happened in 1957 and 1968. Gene segments from avian flu do go into pigs and human influenza viruses. Now, so that is a more likely scenario than suddenly a avian virus jumping into human and then spread efficiently from human to human. That, I think, is relatively less likely. And then now we have coronavirus pandemic. Last time we are fortunate because that virus, the viral load peaked at day 10 after symptom onset. But this time we are unlucky because it peaked very early on due to all this interferon, etc. Now, so we really have to consider SARS-CoV-3, SARS-CoV-4 as future pandemic agent. And I do believe the strategy of Professor Xu Bojian in having the pan-sabbatical virus vaccine is actually very important. Now, going down further, there are many respiratory viruses that is now affecting human. Parainfluenza, adenovirus, all this metapneumo and respiratory syncytial viruses and even measles, all these are by respiratory route, which means that it's not impossible that we have a paramyxovirus jumping into human, which may cause pandemic also. But there is one I think is particularly worrying to me is the picornaviruses, the enterovirus, the rhinovirus. Now, rhinovirus is safe because it only affects the upper respiratory tract, but entero can be very systemic. And in 1997-98 Taiwan major outbreak already illustrate that this may be important. And whether there is one day an animal and enteroviruses jumping into human is another important issue that we have to consider. We need a lot of support from government to prepare for future pandemics. If there is no political conviction, there will be no resources, and therefore all these broad spectrum antivirals, diagnostics, or vaccines that we're thinking now would never come into reality. I think that's all that I would like to say.
Thank you, Professor Yuen. Professor Mollick-Pieris, what do you think?
Yes, I mean, I agree with Professor Yuen that I think what we really need to focus on are respiratory transmitted epidemics and pandemics, right? And these viruses that are transmitted by other routes, we can contain with basic public health measures, but respiratory transmitted viruses are the ones that really pose these major challenges. Now, in fact, this question was asked of me in 2019 at a Keystone Symposium, and I gave the answers that my two top concerns for the next pandemic was influenza viruses and coronaviruses. And the reason was that in both those groups of viruses have a track record of repeatedly causing pandemics. I mean, we didn't have the time to discuss it today, but basically we know that over the last 400 or 500 years, at least 2 other animal coronaviruses jumped to humans, and they are now widespread, they're endemic in humans. So coronaviruses have done this before. But of course, there are other potential candidates like the paramyxovirus group, etc. Now, I am still concerned about the sarbecoviruses, as was pointed out, and also by MERS coronavirus. I mean, as I pointed out, I mean, at the moment, we are really missing the boat, I think, by focusing on the Arabian Peninsula, where I think the potential risk from MERS coronavirus may come from Africa. So I think we really need much more work to be done, both at the surveillance level, at the preparedness level, early response level, and the countermeasure level. Over.
Thank you, Professor Pieris. So in the interest of time, I would like to draw both the roundtable and also the meeting to a conclusion. And once again, I would like to express my heartfelt congratulations to the 2 Future Prize laureates, Professor K.Y. Yuen and Professor Mollick-Pieris. And I would like to thank the 4 very prominent panel members for a very, very fruitful discussion. And thank you all for joining us online and also here. Thank you. Thank you.
Thank you, Professor Hong and all the panel members. The Joint Academic Conference has come to an end. Thank you all for joining us. Have a good day.